think my real interest, if I traced it back, was curiosity about the physical world, which is what physics is.
One might imagine, at least, that one's talking about nature when one's talking about the character of physical laws. But I don't want to talk about nature, but rather how we stand relative to nature now. I want to tell you what we think we know, and what there is to guess, and how one goes about guessing.
This is the piece of the equation which describes how the Higgs field acquires its value.
So welcome to our 43rd Physics Matter Colloquium. So this is already over more than 3 years that we have every month so presented and celebrated science across the geographical frontier. So nearly and mainly centered around the Sesame light source in Jordan, but we have been trying as well to transcend the borders by sharing knowledge in the field of discovery tool. So looking at how particle accelerator, but as well light source, neutron source, and scientific case or education is shaped around the globe. So we have connected as well and teamed up with several international organizations so to better serve this scientific community that is around us. So we recall that this live event is organized by the Forum of International Physics, the FIP, at the American Physical Society, so which is a voluntary association of the APS members. And those people, those members, so like Stefan, who is there, and myself, so we're interested in advancing the knowledge of physics and its diffusion across the world. So our live open access scientific expedition are fostering cooperation and are transcending the borders of knowledge for and by developing community. So visit our website where you will find all this material for those precious expedition, and everything is shared and open source. So last month, so we gave, I gave an insight as well of all the different work that we did using Forum to accelerate the global digital world. So with Stefan, so it was like an interesting possibility to share so with you also what we can do digitally and what Forum means as well. So in March, we had the chief of the basic science section at UNESCO, so presenting the Sustainable Development Goal and the International Decade of Science for Sustainable Development. So a lot of follow-up also with those forums that were also connecting from the one in December and January, where Professor Ellis as well had explained a bit more about the complexity of those goals. So first, to introduce the panelists, so my name is Christine Dorf, so I'm working at the European Spallation Source, so in Sweden. And we have also from the FIP, so our editor for the newsletter, so Stefan Kenmo, who is with us. He's from Germany, from the University of Duisburg-Essen. And we have so the pleasure today, so for our guest star, to have live from Sweden as well. So Professor Giovanna Fragneto, so who is the science director of the European Spallation Source in Lund, in Sweden. So this is an ERIC facility under construction, and so it has the ambition as well to be the most powerful neutron source in the world. So Professor Fragneto so has spent 25 years at the ILL, the Institut Low-Langevin in France, in Grenoble. So this is a nuclear reactor which delivers high-flux neutron beam to study the matters in the realm of soft and hard condensed matter, biological, magnetism, material science, nuclear, and particle physics. So Professor Fragneto has obtained her first degree at the University Federico II in Naples, in Italy, and her PhD from the Physical and Theoretical Chemistry Laboratory at the University of Oxford, so in UK. So as for 2016, so she holds the chair Grand Instrument Européen at the University of Grenoble-Alpes. She has been awarded in 2006 for 2006, for the BTM Willis Prize of the Institute of Physics at the Royal Society of Chemistry in UK. So for her research work on biomembrane in 2023, the Danske Wesen Award, so from the journal Colloid, which is also interface science for the outstanding achievement of the colloid and interface science that she did. She's also the editor-in-chief for the European Physical Journal, E, and the author of more than 220 publications in a peer-reviewed journal. And last but not least, so she's an active involved in promoting a better gender balance in science. So we are really honored to have you with us, so Giovanna, and without further ado, so we will have you sharing the screen and start your presentation. Yes.
Okay. So thank you very much, Christine. Thank you very much for the invitation and the opportunity to present this work. As agreed, the plan is to give a flavor of what neutron scattering techniques can do to explore structures, mechanisms at cell membranes. This colloquium is intended really for non-expert neutron users. So if there are experts in the audience, you will forgive me if some of the things look too easy. So neutrons, why do we use neutrons? What are neutrons? Neutrons are, as you all know, subatomic particles found inside atomic nuclei, and they were discovered back in 1932 by Chadwick. They have a mass, and we will see later that this is interesting and why neutron techniques are different, for example, from X-ray techniques or light. They are uncharged, have a lifetime of 15 minutes, behave both as a wave and as a particle, and this is important for the ways we detect neutrons. Their wavelengths are similar to atomic distances, and the energies are similar to the motion of molecules. And finally, very importantly, they possess a spin. So how do we produce neutrons? They can be produced either by fission in nuclear reactors, or by spallation, where we can send a high energy pulsed electron or proton beam on a target composed of a heavy metal, a heavy element, an element with many neutrons. And then, since the process of spallation will create will release neutrons from inside the nucleus of the heavy atom. There are a large number of neutron sources nowadays around the world. So we find sources in the US, both reactors and spallation sources. In Australia, we have a reactor. In Japan, we have a spallation source and a reactor. China. And in Europe, we have a number of both reactors and spallation sources. I'm going to, as Christine said, I've been working for a long time at the ILL, the Institute of Giovanna in Grenoble. So the work that I will present to you has been collected there, because where I work now, which is the European Spallation Source, it's a facility in construction, and we will be able to use neutrons in a few years, and I will show to you the status of the project at the moment. Actually, the fact that neutrons could be used to look both at the structure of material as well as the dynamics It's something that has been found already more than half a century ago, already in the 50s. Bert Brookhouse and Cliff Schul from the Oak Ridge National Laboratories in the US and the McMaster University in Canada demonstrated how neutrons could be used, would follow the same Bragg laws, could be used to look at structure of material and dynamics of material. And for these studies, they were awarded the Nobel Prize in 1994. In the 60s, neutrons were used mainly for physics studies, so were heavily used for looking at magnetism. So the fact that they possess a spin was highly used. And later on, the use expanded to look at hydrogen bondings, phase transitions, also to areas of chemistry. In the 80s, there were the first measurements in engineering fields or geology, biology. The field has become more and more multidisciplinary and varied. And nowadays, with a number of improvements of neutron sources and a number of improvements also in methods of sample preparation, of data analysis, and so on, there are more and more complex systems under studies and lots of focus in the typical societal challenges like work on health, energy, environment, and so on. Why is that? Why neutrons are such a nice probe to study matter? Because of the wavelength range of neutron beams that are very similar to those of X-rays, we can look at structures that can go from fraction of angstroms, so we can have very high resolution structures of materials, up to big complexes, for example, proteins and DNA or protein-protein complexes. So we can study really a very broad range of time of length scales as well as time scales. And the way in which we extract the information is really by looking at the change of direction and speed of neutrons as they pass through the material. When neutron beams interact with materials, by the way they change direction, we can determine the structure, so follow Bragg laws. And if there is an exchange of energy, we can follow up the dynamics of material. What makes them very different, for example, from X-rays is that they interact with nuclei, and they are scattered differently by different isotopes of the same atom. So while, for example, X-rays, the scattering power will increase with the number of electrons in the element, so going up in the with the number going up with atomic number of the element, we have a higher and higher scattering. In the case of neutrons, they are very sensitive to small to light elements. So hydrogen has a strong scattering power, carbon, oxygen, and so on. And different isotopes are scattered differently. And we use a lot this feature to change the scattering intensity of the material. So like in this picture over there, where you have glass balls that are put into glycerol, and as you well know from your physics studies, the glass are visible in air and they become invisible in glycerol because glycerol has the same refractive index as glass. Similarly, we can replace the hydrogens of material with deuterium to remove signals or highlight signals of parts of our system of interest. So we use a lot neutrons when we are interested in the location of hydrogens in material. Hydrogens are not so visible with photons or X-rays. Or when we want to look at biological or softer material that is rich in carbons or hydrogens in light elements. The fact that they are a neutral particle makes them highly penetrating, so we can imagine light elements deeply buried into materials. Here are imaging structures, images of bones and implants, or in this animation here, we see the movement of water up inside the roots of a plant, so we can access easily water or hydrogens into imaging into elements. Also, because they are highly penetrating, we can look at buried interfaces and use very complex sample environments. When we do our measurements, we can look at samples that are in very bulk sample environments. So use, for example, very high pressures or low temperatures, or do in situ measurements. They are non-destructive, so we can work in physiological conditions. We can look at biological material at physiological temperature in solutions at a given pH. So we do not need to have necessarily crystals, and we do not destroy our sample. And finally, since neutrons, as I said, have a magnetic moment and the spin, they are highly used to study magnetic structures, magnetic fluctuations, and so have wide applications nowadays with magnetic nanoparticles. So we do lots of work as well as on magnetic fields. Now, how do we use these neutrons that how do we in practice do experiments? So first of all, I'm going to show to you how from producing neutrons, and I have animations from the ILL on reactors, once they are produced in the core of the reactor, how they are delivered to the instruments. So first of all, they are sent through some tubes, and then they are sent down through guides by reflection to different instruments that will be used to look at structure of materials or dynamics. So when we produce neutrons, which are these colored things in the animation, they will have different energies and times. We can send them through selectors of wavelengths or energies and define a given range of energy of interest for our system. We send them to the sample. They will interact with the sample, which has a given structure. And then they are collected into a detector. Since they don't have a charge, what you would usually do in the detector, you would use the neutrons to, in a reaction, for example, that could be with a gas or another material, that will create a charge, and then the electric charge is the one that is detected, and the signal studies to work out the structure of the material. So since the discovery of neutrons, nuclear reactors have mainly been built for this kind of work. And so there was quite a lot of activity going on, as I said, from the 50s until the 80s and 90s. Many reactors, which are this yellow, these orange lines, have been built. But we have kind of reached the limit of what a reactor can do in terms of neutron fluxes for this kind of studies. And so in the 70s, between the 70s and the 80s, spallation sources started to be created that allowed the production of the spallation mechanisms produces between 20 and 30 neutrons. So for each neutron that hits the, for each proton, let's say, that hits the heavy element, while during fission you produce 2 or 3 neutrons. So spallation sources can produce a much higher fluxes. And we are now building the European spallation source that before the end of this decade should become the the source with the highest flux in the world. It is a project that has been going on for now about 20 years. The design of this source was completed in 2003. Then it was decided in 2009 to site it in Sweden, here in Lund. In 2014, construction started. And we expect to produce first neutrons next year, by the end of next year. And then science later on, and have a fully working user science program by 2027, 2028. So how does it work? We have a linear accelerator. So we have an ion source, which is basically hydrogen produced by electrolysis of water, that after plasma will produce some neutrons that are accelerated over a 600 meters linear accelerator up to nearly the speed of light. They become really very energetic protons that will hit a target. The target is made from tungsten bricks that are inside this rotating wheel. I've lost the animation, sorry. This should be rotating. We have a wheel which is 2.5 meters diameter and several tons weight. Rotates in such a way that when the proton hits the target, it's always in a different place, so there is not overheating. There is a cooling of the lots of heat will be produced together with neutrons, and the wheel is going to be cooled with a helium circulator, and the neutrons will be sent in a similar way as in the reactor case through neutron guides to different instruments. The project is progressing well. The accelerator, here we have a picture of part of it, should be completed by the end of this year. So we will send, say, beam on dump by the end of this year and start testing the entire accelerator by then. The facility has been built with in-kind contributions from several countries. And for example, France contributed a lot to these elements of the accelerator. And this is why we had a visit of the president of the French Republic to celebrate the end of the delivery of these elements in the accelerator. As for the target part, the target wheel that I showed to you is been inserted into its final location that we call target monolith. Lots of work has been going on to make this, to protect it and to host the target wheel. The protons, the neutrons produced will be very energetic, so we will cool them down with what we call a moderator. So also the moderator has been installed. The uniqueness of ESS beside the flux is the fact that it's what we call a long pulse. Long pulse pulses have a length of nearly 3 milliseconds, which is much bigger than what you have at classical spallation source, so like those that we can find in the UK, like ICES, or in US, like SNS or JParc. And the fact that we have this rather long path allows to use parts of the path to shape the path by using what's in our some chopper systems that will allow playing with the path and with its resolution. As for the instruments, we are building now 15 instruments that will be used to look at the structure of materials, so some diffractometers, structure of bulks or low resolution structures with small angle scattering instruments, and I'll show to you soon what this looks like. Surfaces with reflectometers. We have an imaging instrument, Odin. We have an engineering instrument, an instrument for macromolecular crystallography, and 5 instruments to look at the dynamics of matter. And of these, quite a lot of them can be used, and some of them will be exclusively used for soft or biological science. We will cover a large range of structures. So imaging allows to do measurements up to the micron size, and with diffraction, as I said, we can go down to the fraction of angstroms. And we also cover a quite broad energy range in the milli-electron volt regime. We don't yet have all the possible neutron techniques, but we will set up a call for building additional instruments in the future to cover as big as possible range of length scales and time scales. And here I have some recent pictures of the work in progress on the instrument side. Here, what you see are concrete blocks that are used to shield the neutron guides that reach the various instruments. The target is in this area, and the instruments are all around the target. These guides can become as long as 160 meters to reach the different instruments. And this is one of the instruments that has made the most progress so far. It's a small angle scattering machine called Loki. Neutrons come from this side. The sample is in this area. And here we have very large detector tubes to detect the scattered neutrons. That's the status. But this is actually an old picture. It's a bit more complex now of the imaging instrument. The first diffraction instrument, Loki again, and then Bifrost will be an instrument used to look at spectroscopy in extreme environments. Okay, so in my title, I said that I would talk to you what we do with these neutrons with cell membranes. So I'm gonna now to switch from this technical part and tell you a little bit more about work done on cell membranes. So as you know, cells are the smallest living units of an organism. And all the cells have 3 things in common, no matter what type of cell they are, and that would be a cell membrane that protects and regulates the exchange of material from inside to outside the cell, a cytoplasm, so all the elements, the components inside the cell, and DNA. The cell membranes are rather complex assemblies of both lipids and proteins, so that they not only surround cells, but they surround also organelles inside the cells and around the nucleus. And they are composed, they have a scaffold composed of lipids, phospholipids, which are amphiphilic molecules, are these molecules here that have a head group that is, as we say, hydrophilic, likes water, and chains that are hydrophobic, try to escape water. And they would self-assemble in such a way to expose the hydrophilic part to either the external part of the cell or the internal, which are hydrophilic media, and keep the hydrophobic part, the chains, inside. And then you can find different elements like sterols, proteins, and so on. It is a really rather complex composition, and the chains of these lipids can have different levels of unsaturation and so on. So since they are so complex, they are often modeled. They are studied. Their features are studied with model systems, and I will show to you in a minute what kind of models we've been using. But first, I'll tell you why they are interesting, these systems. First of all, they are really very much widespread in our body. They cover a very large area, the area of a football field in our body. The function of many membrane proteins are dependent on membrane composition, on lipid-protein interactions, and lipid-mediated protein-protein interaction. They have a pharmacological interest as a drug transport goes through membranes. They play a direct role in signal transduction. There are a number of modern diseases associated with lipid composition, like diabetes, schizophrenia, Alzheimer's, Parkinson, etc. They have nanobiotechnological applications, and also they have a really fascinating chemistry and physics. So for all these reasons, there is quite a broad interest in these bilayer systems. And they are really suitable for neutrons because they are the sizes of a bilayer is around 5 nanometers, so perfectly in the range of neutrons, and fluctuations are similar to thermal fluctuations. And when we want to study them with neutrons, we usually must create samples that are adapted to the neutron instruments. So typically, if we want to do diffraction studies, we would have stacks of layers well ordered in one direction. We can do reflectivity, look at surfaces on both monolayers and bilayers in solution, so they are more relevant, and they are techniques that allow to they are a lower resolution than diffraction. So you while with diffraction you can have access to the angstrom level features. So with reflectivity you start from a few angstroms, but still have many other advantages and allow to also look at the interaction of different materials with the membrane. And then small angle scattering allows studies in bulk, so on vesicles or bicelles, and is an even lower resolution technique. So what I've been doing for many years in my previous role was to look at model systems formed by a membrane adsorbed on a flat substrate, typically silicon, in water, and separated from the rigid substrate by another membrane, so that I had a fluctuating system in the water. And I've been using reflectometry techniques to look at the structure on surfaces. That is I mean, throwing neutrons or X-rays is like throwing a stone on a surface and seeing how this is reflected from the surface. With neutrons, so just like light, when we have a layer on the surface, can interfere with this layer, and with light, we would have the formation of interference fringes that will change the spacing of these fringes changes with the thickness of the layer. And very similar with neutrons, we are very sensitive to thicknesses of layers down to the wavelength of the beam. So while with light we are sensitive to micron-sized films, with neutrons we are going to be sensitive to nanometer sized. So what we basically do, we send a beam on a surface, we detect the reflected beam as a function of the wave vector transfer perpendicular to the interface. So what we basically have, we have reflectivities as a function of either the wavelength or the angle of the incident beam. So according to what is on the surface, we can have we have different reflectivity profiles that we tend to fit with a model with layers. So we are in the case of bilayers, we will be sensitive to the layer of the head groups of the bilayer, the chains. And we will have reflectivity changing if we have a different composition of the chain, if we have material inserted into the chain. So we are very capable of determining amounts of material on the surface, or in the case of membranes, amounts of material inserted into a membrane or sitting on the top of a membrane. We play a lot with what we call contrast variation with deuterating parts of the system. So when we have a system, we can do several measurements of the same system in different deuterations, deuteration, and analyze our data simultaneously to get a good description of what we have on the surface. And doing this increases a lot the resolution and the uniqueness of our result. Since these techniques with neutrons are kind of low resolution techniques, to be sure, and we do the analysis with model fitting, to be sure that what we are extracting from the analysis is correct. If we do a simultaneous analysis of several contrasts, we increase a lot the confidence in our results. So we've been using this technique with bilayers to do a very large number of things. We've been looking at the effect of temperature on this floating bilayer, the effect of charges, both in the bulk and on the bilayer itself, current, interactions with gene delivery complexes, interactions with nanoparticles. We've been looking at the flip-flop of lipids through the layer, transmembrane insertion, and so on. Really a large number of things. We have also tried to make, as I said initially, the real cell membranes are very complex, so we have increased the complexity of these layers, inserting cholesterol and other molecules. And in the last decades, we've been working on using lipids, not synthetic, but extracted from real membranes that are a better mimic of the membranes. And also there are methods that allow initial deuteration of these lipids. And this deuteration is developed both at the ILL, but also here at TSS in the MAX. And I'm going to show to you, if I have a few more minutes, some examples of work done with this kind of lipids. One is work related to the interaction mechanism of SARS-CoV-2 with membranes. So this is a cartoon of the mechanism. We have the spike protein that interacts with the cell membranes of the host cells and release the viral genome. And what we have been doing, we have been trying to understand what happens at this level at the moment of the interaction. We done, and this is obviously, as you can guess, work that was done during the COVID times. Was the only way we could get in the lab if we were doing measurements relevant for relevant for the pandemic. So SARS-CoV-2 is an enveloped virus, and the viral envelope contains a large fraction of glycoproteins, also known as spike proteins, that play a fundamental role in favoring diffusion between the virus envelope and the host cell. And this protein interacts with the receptor on the membrane, the ACE2 receptor. And we would like to know what's the interaction of the spike protein with the lipid membrane, because if we know the interaction, we can find ways, formulate pharmacological ways to avoid this interaction, to stop the interaction. So we did reflectometry measurements on bilayers formed on solid substrates, both in presence and absence of this receptor, where we used the spike protein given to us by colleagues biologists. We did a number of measurements with the bilayer alone and the spike protein, the bilayer with the receptor and the spike protein. We combined the neutron measurements also with other complementary techniques measurements, and we could evaluate the level of what we found is that in both cases there was a removal of lipids from the membrane in the presence of the spike. This removal was bigger in presence of the receptor. And this is also consistent with work, with other work found in the literature, where lipid pockets have been found in the extramembrane domain of the spike. And the work was published in scientific reports. Similarly, what we did, instead of looking at the entire protein, we looked at the interaction of parts of the protein, what are called fusion domains, so peptides that seem to be involved in the fusion of the virus with the membrane. So we did also in this case a large number of measurements with the separate peptides, and not only reflectometry, but also small angle scattering and others. And we could come up with a model for this fusion mechanisms, taking into account the different peptides, and to give a suggestion of which peptide was mostly involved into attaching to the cell and which one was involved into penetrating and creating the fusion mechanism. Similarly, besides reflectometry, small angle scattering is another technique that is heavily used for membrane studies. And since we are talking about COVID, I thought to show this example from colleagues here in Sweden, who have done SANS measurements on RNA vaccines and use the small angle scattering techniques. So in small angle scattering, what you do, you have a beam, a selector of wavelengths of this beam that you send to a sample. The neutrons are scattered and then are detected in these two-dimensional detectors. And in SANS, the technique of contrast variation is very popular. So what you would do when you have a system of, say, liposomes or vesicles, or like this vaccine composition, you can play both with the duration of your vesicles and also of the water to match out, to remove the signal of parts of your system. So, for example, if you have a system that looks like that, you can you can match out the solution, that is this green part, with the core of your particle. And in this case, the signal will come only from this shell. Or you can match out the solvent with the shell and have signal that comes only from the internal part. So this has been done on this lipid nanoparticle that have become very famous during COVID, that have a complex composition in the shell. They have both lipids, cholesterol. They have this a special lipid called cationic ionizable lipid that is a critical component that concentrates in liver upon intravenous administration of these nanoparticles. And so these colleagues, Federica Sebastiani and Mariana Yanez Arteta from AstraZeneca, did a number of measurements using different duration of both the cholesterol or the lipids or the ionizable lipid in different match contrasts and could determine where there was an accumulation of cholesterol, in which conditions there was an accumulation of material from the outer side of the nanoparticle or the inner side. And knowing where this material goes is very important for optimizing the delivery of the drugs. Another thing that they did, they tried to see what was the effect of a small protein, the apolipoprotein, responsible for fat transport in the body. So they looked at how the composition of these nanoparticles changed in interaction with this protein. I have basically finished. In the next 2 minutes, I want to show to you where we are going to do these experiments in the future at TSS. So we are building a small angle scattering instrument called Loki. You saw the pictures earlier on. That will be a very high flux instrument that will be used for looking at microfluidic systems or very biological samples that are usually very weak scatters. Will be used to do kinetic measurements and in situ measurements and non-equilibrium studies or very small samples. For surfaces, we have 2 reflectometers. One is STIA, it's very special. It's going to be specialized to use very, very small samples. It has a peculiar guide that allows to basically transport without losing with divergence the beam to a very, very small surface. As well as Freya, that is a horizontal reflectometer, very, very fast, and where all this kind of studies in soft matter and biology will be performed. And with this, I'd like to thank you for your attention and invite you to consider using neutrons for your research.
Thank you very much for this extremely comprehensive presentation. And as well, so neutron and light source as well with our neighbors in the max source. So it was extremely complementary as well to the presentation that we had so in April 23, so from Chalmers University as well. So I think it's perfect to have an overall view on what we could do in Sweden and beyond, so in Europe. So thanks a lot. Should we, so we invite everyone to ask and to put the question and answer, so box, you put your question there. If you raise your hand, we could give you as well, so live, the possibility to answer, to ask the question. So we have a first question, so by Wilson Stairs, so asking, so in the set of foil, so detailed in the production process for creating the neutron source, one of the earlier foils showed, so was what happened? So what happened to be blushed colored liquid? Was it water, or was the blush coloration being produced by the Cherenkov radiation? So maybe if you can come back to that.
Okay, so well, yeah, the very first slides, yes, is Cherenkov radiation. No, it's the reactor, you mean?
The reactor, yeah.
Sorry, I need to go back a long way. Here.
Think it
this one. This is Cherenkov radiation.
This is the Cherenkov, but even in the, maybe it was the other one. So there, yeah, clearly that the Cherenkov, and the next one after that, there, maybe because we see some.
That's the water pool around the reactor. So these are, let's say, animation, while the one that I showed here, this is a picture. So sorry, it starts this thing, but this is a picture. You see, that's a picture. But the rest is animation. Yeah.
Yeah, very good. And indeed, so Wilson said that that was that one. So I think that's good. So with so that answer, we have also Coyote, so that is also asking. So after thanking you for this presentation, so please, I want to know, so if the studies are best done so with purified biological material or some fresh sample. So looking at, for instance, isolated human sample, how big as well maybe the size of those sample, and how much sample preparation is needed for the neutron studies, so sample environment as well.
Yeah, so many questions. So yeah, but ideally we want to work with as pure as possible. These techniques are very sensitive to contaminations. So the more pure is the system, the better. And sample preparation really very much depend on the technique that you are using. To make bilayers, you can, with natural lipids, you make them by spreading from vesicles. So you put in contact the surface with a solution of vesicles, and you should play a bit with temperature or with some parameters, salts, etc. And then the vesicles will just go on the surface, and you wash out and you make a bilayer. For small angle scattering studies, you can you make your sample either with sonication or extrusion. So you need to optimize. You need to become a little bit expert sometime, but it's a very reasonable time scales. As a sample, yeah, you make in a couple of hours or something if you know what you are doing. If you are, skilled in doing the extrusion or the sonication or this spreading, you have to clean the surface and so on. But it's a matter of hours.
And maybe also in that same register, the importance as well, the influence of the long pulse, or looking at, for instance, by comparison to what they could do with SNS or GPOC.
But the interest of long pulse is this fact to play a little bit. Maybe I can go to the slide to play with the resolution. And so to play, use either you can use the entire length of the path for some measurements and have a very intense beam, or you can kind of work with the resolution by selecting a given fraction of wavelengths in your path, a given fraction of time in your path. It's a matter of flexibility in being able to have instruments doing different things.
Very good. So it will be some interesting complementary measurement as well. Huh? So as soon as we have BIM. So with, and as well, I think there was some, and maybe we can share the presentation of the publication. So that was a question from Sabrina in Terminator in Hungary, asking, so if you could share as well the presentation, the papers for the COVID. So were there several of them, and maybe we could as well add them in the material together with the slides.
Yeah.
So that would be really interesting. And I think that what would be also interesting, because that was in July 2021. So we had also from Adayona a presentation while we were in Israel. So I had in the material as well shared the presentation that she gave between DSS and Max 4. And I think it was also very interesting to see for the ribosome in that case, but how or the life science aspect can be tackled with neutron and light source. In her case, it was mainly light source. And the development.
She's done a lot of work on viruses, synchrotron radiation, but yeah, a long time ago used also a little bit of neutrons.
Yeah, indeed, a neutron was, and in terms of indeed the evolution. So now we have so high flux. So this is also the interest. And also speaking about complementarity or asking with, because I think I remember one of the presentation at the beginning was showing the plant and the interface as well. So that was with neutron, isn't it? Because
Yeah.
I thought I saw this as well when we had a presentation from Sirius in August 2021. So could it be that it was also some complementary measurement? Yeah, this one, exactly. Or you also done with
Usually we very often do complementary measurements of neutron and X-rays because with you have the you have much better resolution with X-rays, but you see the metallic part, the heavy part. If you have some light elements, you see it less. Now in this case, it is not excluded that you can get images of the roots with some accuracy with X-rays. But what's nice with neutrons is how sensitive you are to the movement of the water inside.
Yeah, focusing on the water, and that's why the very nice complementarity. And I think there was a lot of development as well with the case of the sesame, looking at how with the agriculture as well, there could be some development. So very interesting to see this complementarity. So thinking about chemistry, we have a chemist expert as well with us, with Stefan. So Stefan, do you have any questions?
No, fine. So, I mean, neutrons are actually very important for us because, with particularly when we deal with a catalytic system, right, where aqueous solvation, right? So the character of the aqueous environment is very capital, right, to drive the chemistry and so on. So we need to trace the hydrogens, right, interfacial OH hydrogens and how they diffuse inside the the catalytic system. And then neutrons are very important, right, to capture, I mean the motion of these hydrogens and, for example, follow hydrogen evolution pathways and so on. So now I had a question actually about how to distribute the neutrons. So I see the ESS more like a very big bank, right, of neutrons in the future. And I wonder how people will, analyze the samples, throughout Europe. So will people send, I'm not an experimentalist, this is why I'm asking this stupid question. So if everybody in Europe want to use ESS, right, how are you going to operate there? So is it people will ask for beamings, like people doing DAISY, or what? How, like I see it like a central bank of neutrons. How will it?
Yeah, it's not as simple as that. It is a European facility that is at the moment is funded by 13 countries. And so what will happen is that when we produce these neutrons, a good part of these neutrons will be distributed in the member countries. Which means also that if people are not in the member countries are Sweden, Denmark, France, Italy, Germany, Spain, Hungary, but I can show them to you. So Switzerland, UK, a big part of Europe is member countries. So no member countries can collaborate with member countries, or we will have some time, very small amount of time reserved for excellent science, so that there will be some opportunities for non-member countries, but it's very limited. The idea is that researchers will submit proposals, so they will submit very likely twice per year. They will submit a proposal, say what they want to do, on which instrument, how, and show that they can do it. in the large majority of cases, we expect if the beam time is awarded by expert committees, this proposal should come and do the experiment here. In some cases, they can send the sample and we can do it for them, but this will be not the vast majority. There are cases when you can send the sample, and in other cases, you really need the PI to be there and help with the experiment. And then we will help with the experiment, provide sample environment, and provide help with the data analysis. It's going to be a collaborative effort with us and with the member countries and exceptionally outside. Yeah. It's a long answer.
Thanks a lot.
Yeah, but that's I think really interesting and important as well. So we have also in the comments from our colleague at the SNS, so with Jason Gardner. I don't know if you know him, but indeed he was so saying indeed how important as well large scale facilities are. So they are very incredibly so useful. So for our research and as a user relatively cheap. So speaking about beam time, so and one of the question as well, so which is related to industry. So indeed, beam time industry, they're going to be a portion as well of this beam time related and focusing on industry who will pay as well more cash. So do you know the percentage or what we could expect? And if we are intending maybe to develop that, that could be a good way to also get
yeah, definitely the society expects us to contribute to applications, and this we will do it in different ways. So there might be companies and the model that works in other places as well today. Some companies collaborate with academia or with us, and they are interested in doing research that is not necessarily proprietary research. They are happy to publish their data, and we will encourage collaborations in this sense. And they will have access to beam time like other users, putting down proposals. We might have panels a bit more specialized in this area, which are more of applications. So the criteria are slightly different in fundamental research and application research. There will be also possibility to buy beam time. So we are working now setting up costs, and the amount is not quite yet defined, but could be between 3 and 5%, but we are working on that. We don't exclude, we have received expression of interest from companies to finance, to build instruments. It's a model that is used quite a lot in JPAC in Japan. We are exploring this opportunity, which has some interest and some drawbacks. At the moment, it's a very exciting moment because we are building up a lot the way it's going to work. But definitely, we are going to push for industrial use.
Yeah, that's the important thing for applying directly as well to the society. And as well, one of the important aspects is the PhD students as well, who will work on industrial case in that sense. So industry is always there in substance through the university. And maybe this is a phase 2. So while we will have first the beam, so it will be interesting to build up as well more in capacity for students to obtain the PhD.
I mean, as you see in my concluding slide, I have examples of here is pharma and batteries and some material. We are going to have PhD programs. At the moment, they are in these original plans that we are drawing at the moment. We want to have an opening for next year, and we would like to have students here able to profit of some neutrons. So we will open the call not before end of next year. At the moment, we are focusing more on academia, not so much on industry, but I know, for example, at the ILL ESRF, there are programs of PhD in combination with companies. So it's not excluded that we can become part of this kind of programs.
Yeah,
So,
that's
Europe grants, for example, with Luxembourg company.
Christine, there is another question in the chat.
but just on that topic, if in terms of figure, because I remember it was 40% in terms of this program as well, the return from industry with student in the case of ESIF and ILL. So it will be quite a very, very impressive way. So yeah, thanks on that.
If we could go in one of these programs like the innovation program, ILL ESRF, that's an interesting one. But the problem is that companies work fast and want the thing done quickly, and at the moment it's really difficult to engage with companies because the facility is not running. We need to commission instruments. We need to know what are their performances. It's not like SRF, ILL, or places that have been running for a while where they can make promises to companies that can be reasonably maintained, that they know what's their flux, they know what are the capacities of their instruments. For us at the moment, it's really premature to think at companies. They need, But we are happy to collaborate with companies to set up programs and start using other facilities just to get them involved, and then eventually come to ESS in a few years' time.
Yeah, that's really an interesting and exciting future. Indeed, because the question that is on the question and answer is very interesting as well. So from Huatawa, so Kundu, who is asking about the neutrino. So can neutrino scattering be used instead of neutron scattering? So as neutrino and neutron, so as well. And why not? And maybe to mention indeed the program for neutrino, even if it's not exactly in that way.
Yeah, well, there is some interest to make a neutrino factory or to make neutrinos, but at the moment it's not in the plans of ESS. ESS has been funded for neutrons, so we don't have plans. There are some ideas, and we might go out with a call to detect neutrinos. But to make neutrinos, there are no plans at the moment. We need first to deliver neutrons. It's not excluded in the future. I know that there's quite a bit of interest, but it's not our top priority at the moment.
Indeed, we first need the neutron, and then also with the ring. So if we would have to build an additional infrastructure with the accumulator.
Yeah, it's a really big program. There are ideas, but really in the future we need now to concentrate on the near future.
Exactly. So useful case, but still opening as well with industry, as you said. So I think that we have an excellent.
There's lots and lots of potentials, and lots of things that will be done. But we at the moment we really feel the urgency of starting up. We are very impatient to having first neutrons, commissioning first instruments, getting instruments online.
Yeah. Excellent. So thank you so much for this presentation. It was very, very interesting for having a full review of those capacity. So we will, let's see, so thanking you as well. So no more questions. So in the chat box, you can always contact us and ask more questions. So we will be happy to to answer. And we will put the slide available as well on the web, like the recording, which should be available next week. And we will so also mention, so for next month, so we will have the next physics matter colloquium, which will be a forum. So the forum phase, which is like a follow-up as well from the first one we had in January. In this case, so we'll try to make it shorter with less people, but looking at how sustainable development so can be answered as well by decomplexing the complexity in one sense. And as you have seen as well in our series, we try to tackle this sustainable development, environment aspect, and the life science. So I think this is one big project as well for having more of such type of presentation like Giovanna gave today. So thank you very much to have attended this presentation. So, and we'll hope to see you soon, and we'll stay tuned. So thank you very much, everyone.
Bye-bye.
Thanks a lot.